Good morning, good evening, good afternoon, good night. As per the situation, you can take one of them. Today, I'm going to explain you something about genetics once again. This is the second video for the genetics for class 12. In last class, I have explained you some contrasting traits, 7 contrasting traits of pea plant. Today let me go with some important topics, that is terms as well as definitions. So first we should understand what is genes. Genes is a segment of DNA that codes for particular character. Genes is a segment or piece of DNA that codes for particular character. So similarly, we should know what is alleles. Alleles are alternate form of same gene for same character. Alternate form of gene for the same character, that is called as allele. For example, capital T are the alleles, capital T small t are the alleles, or small t itself are the alleles. So when we are comparing two genes together, then we can call that as allele. Like we say couple, like that here it is alleles. So when we talk about other than that, we should understand what do you mean by monohybrid cross. When you say monohybrid cross is a cross that is made to see or study the transfer of single trait from parent to its offsprings. From parents to offsprings, if single trait is transferred, then that is called as monohybrid cross. For example, out of 7 contrasting characters that we studied, one character transferring if we are studying, then that is called as monohybrid cross. For example, if you take the height of the pea plant, there is one character. In that you will find capital T as dominant character and small t as recessive character. So and when we are crossing them, then the resulting will be hybrid. That is, it is having one capital T and it's one small t. Now on the basis of this, we can make a law that is called as law of dominant, where we can see in an hybrid, when you study monohybrid cross in an hybrid, one of the character is dominant and is expressed, whereas another character is recessive and is not expressed. The expressed character is written in capital letter, whereas the not expressed character is written in small letter. So this is called as law of dominant. So along with this point, you can also add 2 more points. You can say that Mendel called genes as factors. Mendel called genes as factors, and factors always occur in pair. So these are the 3 points that you can say in law of dominant. Whereas when you talk about monohybrid cross, we had one more law that is called law of segregation. Like I told, when we are taking 2 parents, one capital T and one small t, and when we are trying to cross them, we need to take out the gametes, and that gametes will be capital T as one single letter as one gamete and small t as single letter as another gamete, and when we are fusing them, we are getting a hybrid. So here we know that while formation of gametes, the factors are getting separated from each other. So 2 capital T's are getting separated. Similarly, 2 small t's are also getting separated. So that's forming a law called law of segregation. Segregation meaning separation. So according to the law of segregation, while formation of gametes, factors get segregated, and one factor enters one gamete and another factor enters another gamete, and that is called as law of segregation. And law of segregation is also known as law of purity of gametes. This is about monohybrid cross. Okay, and in monohybrid cross, other than this monohybrid cross that I am talking about, we have some more terms that we should learn, and that is one of them is Punnett square. You know Punnett square. Punnett, it is a scientist name. He had discovered a square so that he can represent this flowchart in simple form. So for example, in a Punnett square, we write capital T towards one side, small t towards another side, and we mix the gametes and we write the answer. And that's in the form of square. That's why it is called as Punnett square. Don't make a rectangle, make a square. Okay, so and also along with that, we should also know some more points like test cross. So we know that when a phenotype character is given to you, it will be very hard for you to find out whether it is homozygous or heterozygous pure. So to find this, whether the given dominant character is heterozygous or homozygous, the cross that we make is called as test cross. In order to find out whether the given character is heterozygous dominant or homozygous dominant, the cross given is made is called as test cross. For example, capital T small t is given to you as a tall plant, but you will not know its genotype is capital T small t, and unless you do the test cross. So same thing, if it is given capital T to you, and unless you do a test cross, you will not know that you got capital T. So during test cross, what we do is, whatever the character is given to us, that one we is crossed with recessive parent. When we say recessive parent, it is small t, and then when you are crossing, if the result contains any of the recessive character, then the given dominant parent is heterozygous, and if it does not contain any recessive character, then the given character is a homozygous character, that is capital T. So in that way we can just find out what is test cross. For today it's okay, this much, and in next class I will be dealing with codominance, incomplete dominance, sex determination, and many more. Be tuned, and if you haven't subscribed my channel, you can subscribe, and I will help you. Whatever doubts you have, you can just put on in comments, and if you want live session, I can give you live session too. Thank you, bye bye, have Subharatri, good night, best of luck, do your exams best, bye bye.